Mendel was the son of a peasant farmer and was born in Czech Republic in 1882. He went to the University of Vienna, wherein he studied botany and learned the scientific method of his work. Prior to Mendel, heredity was regarded as a blending process, and the offspring were essentially a delusion of the different parental characteristics. In 1886, he published the Experiments in Plant Hybridization, in which he established his three principles of inheritance. Let's leave it to Joanna to enumerate the three.
First, principle of dominance. One allele must another. One allele was dominant over the other in the F1 generation. Second, law of segregation. The law of segregation states that for any trait, each parent's pairing of genes or alleles split and one gene passes from each parent to an offspring. Which particular gene in a pair gets passed on is completely up to chance. When gametes are formed, the pairs of hereditary factors or genes become separated, so that each sex cell, or which we call egg or the sperm cell, receives only one kind of gene. Third, law of independent assortment. The law of independent assortment states that different pairs of alleles are passed into the offspring independently of each other. Therefore, inheritance of the genes at one location in a genome does not influence the inheritance of genes at another location. Remember this. Members of one gene pair segregate independently from another gene pair.
Mendel carried out breeding experiments in his monastery's garden to test inheritance patterns. He selectively crossbred common pea plants with selected traits over several generations. He tried to repeat his work in another plant, but didn't work because the plant reproduced asexually. His work was largely ignored for 34 years until 1900, when three independent botanists rediscovered Mendel's work. Have you ever wondered why some of your classmates have similarities with their parents? Try looking at your classmates and ask them, are you more similar to your mom or your dad? Well, some people would say that they're a mixture of both. But there is a scientific explanation to these types of questions. That leads us to a scientist named Gregor Johann Mendel.
A dihybrid cross illustrates the potential results from crossing individuals that differ in regard to two traits. In this case, the parents, or pea generation, differ in both pea color and shape. A plant homozygous for wrinkled green peas is crossed with a plant homozygous for round yellow peas. Because both parents are homozygous for both traits, only gametes coding for wrinkled green peas or gametes coding for round yellow peas are produced. A diagram of the cross between two members of the F1 generation, called a Punnett square, is shown here. When the gametes from these pea plants are combined, all of the offspring in the F1 generation are round and yellow. This is because the traits for round and yellow are dominant over the traits for green and wrinkled. Because all offspring within the F1 generation are heterozygous for both traits, both the males and females can produce 4 different gamete combinations: round yellow peas, wrinkled yellow peas, round green peas, and wrinkled green peas. A Punnett square is created to determine the F2 generation that results from this cross. Of the offspring produced within the F2 generation, 9 are round yellow peas, 3 are round green peas, 3 are wrinkled yellow peas, and 1 is a wrinkled green pea. In other words, a 9:3:3:1 ratio exists within the F2 generation. Mendel was able to use this observation to determine that factors assort independently of other pairs of factors. This has become known as the law of independent assortment. To determine the principles of segregation, Mendel crossed two plants that differed in one phenotype. In the cross shown here, the members of the parent generation, or P generation, differ in only one trait, which is color. The purple pea plant can only produce gametes that code for more purple pea plants, and the white pea plant can only produce gametes that code for more white pea plants. A diagram of the cross between two members of the F1 generation, called a Punnett square, is shown here. All of the possible types of eggs are lined up on one axis, and the types of sperm are on the other axis. When the gametes from these pea plants are combined, all the offspring in the F1 generation are purple. This is because the trait for purple plants is dominant over the trait for white plants. Two heterozygous purple plants from the F1 generation can be crossed to produce an F2 generation. In the F2 generation, 3 purple pea plants are produced for each white pea plant. Therefore, purple and white phenotypes are produced in a 3:1 ratio. A Punnett square is often used to better understand the patterns of inheritance associated with Mendelian genetics. Remember that each parent in a genetic cross is diploid, meaning that they have 2 copies of each trait. During meiosis, these traits separate into individual gametes, either egg or sperm cells. In a Punnett square, the outside of the square represents the possible gametes that may be produced by each parent. In our example, a plant that is heterozygous for flower color produces gametes that either have the purple or big P allele, or the white or little p allele. The area within the Punnett square represents the possible zygotes, or offspring produced by a combination of a specific egg or sperm cell. Note that we cannot predict which egg cell will be fertilized by which sperm cell, so a Punnett square indicates all of the possible combinations. A Punnett square not only allows us to visualize the potential offspring from a cross, but also allows us to calculate the probability of obtaining an offspring with a specific phenotype or genotype. Notice that, assuming both parents are heterozygous, that there is a 3/4 chance that the offspring will have the dominant purple phenotype, and a 1/4 chance that the offspring will be white. Punnett squares are useful for quickly visualizing the results of a single trait or 2-trait cross.
Gene, a unit of heredity. Genome, the entire set of genes in an organism. Allele, 2 genes that occupy the same position on homologous chromosomes and that cover the same trait like flavors of a trait. Locus, a fixed location in the strand of DNA where a gene or one of its alleles is located. Genetics, a branch of biology that deals with heredity and variation of organisms.
Homozygous means having identical genes, one from each parent for a particular characteristic. Heterozygous means having 2 different genes for a particular characteristic. Dominant is the allele of a gene that masks or suppresses the expression of an alternate allele. The trait appears in the heterozygous condition. Recessive is an allele that is masked by a dominant allele. It does not appear in the heterozygous condition, but only in the homozygous. Is the genetic makeup of an organism, while the phenotype is the physical appearance. That's it for today, guys.
Thank you for watching. Bye-bye.